Hello, very good evening children. So today we'll be discussing about the DNA fingerprinting, which is a major application of human genome project. Okay, so the DNA fingerprinting, this technique was developed based on certain facts that we have derived from the human genome project. So what are the two major pieces of information of the HGP that led to the development of this technique, right? So there are two bits of information here. Number one, according to HGP, we came to know that 99.9% of the human genome, right, is the sequences are the same. The base sequences are the same in every individual, right? And we differ from each other only in terms of this 0.1% of the sequences is only the difference, right? So that means it is this small percentage of variation. It is this 0.1% of variation in the sequences that makes every human unique. It makes every human unique, right? Alright. So now this is the major information that we got. And the second major information that we derived from HGP is that out of the total genome, about 2% of the genome, 2% of the genome is coding for proteins. Coding for proteins. Then what is happening with the rest of the 98% of DNA? Well, so this 98% of DNA, suppose we assume that this 98% may, some of the DNA is coding for, rRNAs, tRNAs, regulatory sequences, and so on. Majority of this DNA is not having any direct coding function. Okay? Majority of this DNA has no coding function. Now, this DNA, majority of it is non-coding, right? And within this non-coding sequences, they have identified that there is a lot of repetitive DNA. Majority of the genome is made up of non-coding sequences. Now again within this non-coding sequences, most of it is made up of within this non-coding sequences, again you have two different types of, one is the repetitive DNA, two categories. Repetitive sequence DNA, and the second category is non-repetitive DNA. However, out of the whole thing, repetitive DNA will make up the major portion, and we are interested in this repetitive DNA. Our interest lies in repetitive DNA that we are using as a probe for DNA fingerprinting, right? Is this clear? Now, why the name repetitive DNA? This DNA is made up of repeated sequences. It is made up of stretches of DNA sequences that are repeated for many times. DNA sequences that are repeated for many times, right? Okay. How many times? It can be 100 times or it can be 1000 times. It depends. Now, what is the use of this repetitive DNA if you ask me? These repetitive DNA, as they do not have any direct coding function, they have no direct coding function. But then what is their use? According to scientists, they say that they can be useful to understand the structural dynamics. They can be helpful to understand the structure of chromosome. Okay. They might also be helpful to, shed light on the evolutionary process of the DNA over a period of time. Okay, and it is this repetitive DNA that we are using as a basis for DNA fingerprinting and gene mapping. And it also shows a very high degree of polymorphism. It shows high degree of polymorphism. And it is this repetitive DNA that is the basis for the DNA fingerprinting as well as human gene mapping. Basis for two things. So this was I think last year question also. What forms the basis for DNA fingerprinting? High degree of polymorphism is the basis for DNA fingerprinting and also for human gene mapping, right? Now,